Hello everyone, my name is Larsa Alobaidi and today I will be presenting a study titled Gut Microbiota Dysbiosis and Vitiligo, the role of hippuric acid in oxidative stress. This presentation is a part of my work for the microbiome class and it explores how imbalances in the gut microbiome, also called dysbiosis, may contribute to skin autoimmunity. Specifically, we will focus on vitiligo, which many of you know is a chronic condition that leads to the loss of skin pigments. We will look closely at a particular microbiome metabolite called the hippuric acid and see how it may link gut health to oxidative damage in the skin. Over the next 30 minutes, I will walk you through some background concepts about vitiligo, oxidative stress, and the gut microbiome. The study's main goals and research questions, the methods and models used, the key results, the figures, and finally, what does this all mean for the future of treatment? So let's get started. To begin, the bottom line takeaway of this study is that gut microbiota dysbiosis may play a casual role in vitiligo. By promoting oxidative stress. While the idea that gut health affects the skin, sometimes called the gut-skin axis, has been proposed in the past, the exact molecular mechanism behind this connection wasn't clear. This study aimed to fill that gap by identifying a specific microbial metabolite, which is the hippuric acid, as a key link between unhealthy gut microbiome and increased oxidative damage in vitiligo. So to do this, the researchers used an integrated approach. They gathered data from vitiligo patients and also used mouse models, combining multiple omic tools like microbiome sequencing, metabolomics, and transcriptomic to get a broad, detailed picture. They showed that hippuric acid levels were elevated both in patients and in experimental animal. So more importantly here, they demonstrated that this metabolite could directly promote oxidative stress in the skin. So this is important because it suggested that gut microbes are not just pathogens but active drivers of disease progression in vitiligo. These insights open the door for new treatment strategies that focus on restoring gut balance or blocking harmful microbial products. Before we dive into the experiments themselves, let's step back and look at the biological concept. The gut microbiota, the trillions of bacteria are living in our intestines. It's a bacteria, viruses, parasites, a lot of living there in our intestines. They play essential roles in digestion, metabolism, and immune system regulation, and more a lot. More recently, researchers have discovered that gut microbiome also produce metabolites, small molecules that can travel beyond the gut and affect distant organs, including the skin. So meanwhile, vitiligo is a chronic autoimmunity disease where melanocytes, the cell responsible for skin pigment, are damaged and destroyed. One of the main factors contributing to this destruction is the oxidative stress. Reactive oxygen species, they called it ROS, can damage melanocytes' DNA, protein, and membranes, eventually leading to their death. Although recent studies have hinted at a link between gut dysbiosis and skin autoimmunity, the precise mechanisms behind this connection in vitiligo have remained unclear. This is the knowledge gap that the study set out to address. How exactly may gut-derived molecules trigger or worsen oxidative stress in the skin? As the researchers noted in their abstract, the complex interaction between abnormal accumulation of ROS in the skin and gut microbiota has remained unclear. This statement highlighted why this research matters. We know that vitiligo involves oxidative stress, but we don't fully understand where this stress comes from or how it's become chronic. Existing treatments like topical corticosteroids or immunosuppressants can help slow pigment loss, but they don't target any root cause. If gut microbiota and their metabolites are part of the problem, then modifying the gut environment could offer an entirely new way to treat vitiligo. So, this study doesn't just fill a scientific gap, it also points toward promising therapeutic possibility like probiotics or metabolic inhibitor. With this context, the researchers set out to answer three core questions. 1. Does gut microbiota dysbiosis contribute to vitiligo progression by targeting oxidative stress? 2. Is hippuric acid the microbial metabolite specifically responsible for driving this oxidative stress? 3. Could this connection help explain how gut health influences autoimmune skin condition more generally? So by answering these questions, they hope to reveal a novel gut skin mechanism and identify a new treatment targets. To tackle these questions, the team used a multi-omic approach. They began by collecting stool and blood samples from vitiligo patient and healthy control to assess microbiota composition and measure the serum metabolites. They then used a mouse model of vitiligo, transferring gut microbiota from patients into mice to see whether this induced disease-like features. Key techniques include 1. The 16S RNA sequencing, to analyze the composition and drives diversity of gut bacteria. To metabolomics to detect and quantify serum metabolite, where hippuric acid emerged as a candidate of interest. 3. The RNA sequencing to examine gene expression in skin samples, especially gene related to oxidative stress. Histology and Ross assays to visualize damage and measures oxidative stress in the skin. Direct metabolite demonstration to test causality by injecting hippuric acid into mice and observing outcomes. This integrative design allowed the researchers to move from correlation to evidence of causation. Let's briefly highlight what each method brought to the study. So, 16S RNA sequences clarified which bacteria were more or less abundant in the vitiligo. Metabolomics measured changes in small molecules traveling in the blood. RNA sequences provided snapshots of skin gene activity, especially those involved in inflammation and oxidative stress. Histological staining showed tissue level changes, loss of melanocytes, and damage to skin too. So, the microbiota transplantation proved that transferring dysbiotic gut communities could induce vitiligo-like feature in mice. Direct hippuric acid injections confirmed that this metabolite alone was enough to increase oxidative stress and depigmentation. By combining these tools, the researchers built a strong case for a gut-skin connection mediated by hippuric acid. So let's dive in the figures and we will talk about them one by one. Let's start with figure 1. This figures shows gene expression in skin from vitiligo patient using 2 large databases. They identified many genes, either more active or less active in lesional skin compared to nearby healthy areas. The affected genes were linked to immune activation, oxidative stress, and distributed depigmentation. This confirms that oxidative damage is a hallmark of vitiligo and sets the stage for connecting these changing to get derived metabolites. This is the figure 2. Here mice were engineering to develop vitiligo by introducing melanoma cells and regulatory T cell. Over time, their skin lost pigments, similar to human vitiligo. When researchers examined the skin, they saw more immunocells and fewer melanocytes. Genetic analysis revealed altered expression of genes involved in stress and inflammation. This model allowed testing whether gut microbiota or metabolites could worsen these effects. Together, these figures here, like in figure 2, confirm that the melanoma trait induced vitiligo mouse model replicated many of cellular, immunological, and transcriptomic features of human vitiligo, making it a powerful tool for mechanistic study and therapeutic development. For figure 3 here, in this part some mice reserve antibiotics or reduce gut bacteria before vitiligo was induced. Mice treated with antibiotics had less oxidative stress and healthier skin structure. This suggested that gut microbes contribute to damage, supporting the idea that microbial metabolite may be involved. So the overall of these figures here for the figure 3, these findings show that gut bacteria strongly influence oxidative stress, mitochondrial damage, and gene activity in vitiligo, and removing them with antibiotics can help reduce these harmful effects. So, the figure 4. This figure shows experiment where the researchers manipulated the gut microbiome by co-housing, fecal transplanting, and probiotic. All these interventions changed how much pigment was lost. Mice given healthy bacteria or probiotic shows less pigmentation over time, reinforcing that the gut microbiota influences disease severity. So the overall here, this result shows that vitiligo is linked to changes in gut microbiota diversity and compositions, and that restoring or modulating the microbiome, whether through co-housing, fecal transplant, or probiotics, can help protect against pigmentation in this mouse model. So for here, the figure 5, like target metabolite revealed that vitiligo mice had higher levels of certain metabolites in feces, blood, skin. Notably, uric acid levels were elevated in all compartments. When antibiotics were given, these levels dropped, again suggesting the gut as the sources. So overall, this result shows that vitiligo disrupts metabolite profiles across the gut, blood, and skin, and that the gut microbiota plays a major role in shaping these metabolic changes. So, here the figure 6. Injecting uric acid into mice caused higher oxidative stress and more pigmented loss than other tested metabolites. This strongly supports its role as an active driver of skin damage. So overall, these experiments shows that uric acid a gut-derived metabolite can accumulate in the skin, increase oxidative stress, and worsen depigmentation in vitiligo. This highlights a direct link between gut metabolites and skin pathology. Lastly, the researchers examined gut barrier function. Vitiligo mice had thinner intestinal walls and higher permeability, meaning molecules could leak into the bloodstream more easily. Hippuric acid moved from the gut to the skin, where it is bounded directly to protein like the NOS2 and MAPK14, those key mediators of the oxidative stress. This final figure ties together gut dysbiosis, leaky gut, and direct molecular damage in the skin. So, in the discussion, or let's say in the summary, the RNA sequencing data confirmed that vitiligo skin has higher oxidative stress and immunoactivation. Combined with microbiome and metabolomic data, we now see a clear story. Gut dysbiosis leads to increased hippuric acid, which leaks into circulation, binds to the skin protein, and drives oxidative stress that damages melanocytes. This mechanistic link helps explain how gut health can influence autoimmunity skin disease. So basically here is the gut dysbiosis lead to increase the hippuric acid. Hippuric acid goes to the circulation, bind to the skin protein, drive oxidative stress that damage the melanocytes. So, conclusion. This study presents strong evidence that gut microbiota imbalances contributes to vitiligo progression through hippuric acid by combining multiple omics techniques. The researchers created a detailed model of how gut microbes, metabolites, and the immune system interact. These findings could inspire a new treatment, such as probiotics, dietary interventions, or inhibitor for harmful metabolites to restore balance and protect melanocytes. So, let's start with the question and asking some question here. So to wrap up all this, I would like to open the floor to discuss with a few questions. Number 1, how strong do you think the evidence is that hippuric acid drives oxidative stress in vitiligo? Number 2, what are your thoughts on the value and the challenges of combining microbiome, metabolomics, and transcriptomics data? Number 3, could we influence hippuric acid levels clinically through diet or supplementation? Number 4, are there other autoimmunity disease where these gut skin mechanisms may be at play? So do you think it's better to target specific microbes or the metabolites they produce? So all these questions here we have, or more, or maybe you have more than this. We can discuss those to find the answer that may help us. Finally, these are the references I used, including the awesome main article and supporting research on the gut-skin axis. I use another research just to have more information about this disease and about the microbiome in general. So thank you so much for listening. I hope this talk give you something new to think about, or at least made you look at your guts a little differently. I would love to hear your thoughts or answering any of the question we asked, or if you have more question, we can address in the chat. That would be awesome to discuss together. Thank you so much.